I'm speaking with Harmit Malik, a member in the Division of Basic Sciences at the Fred Hutchinson Cancer Research Center. Thanks for joining me today, Harmit.
It's a pleasure.
Today we're going to talk a little bit about what you do for the chapter on evolution in our textbooks. But before we get into that, I'd like you to tell us a little bit about where you were born and raised and educated.
So I was born in Agra, the city of the Taj Mahal, but I actually don't have any memories of that. I grew up mostly in the city of Bombay. And I grew up in a family that was made up of business people and engineers, but they really valued excelling in education. So I sort of went on that path and ended up in engineering school. It's sort of an interesting story. I was very keen on medical school for most of my schooling career, but I was underage by 3 days, as it turned out. So I got admitted into the medical school, but I couldn't actually join that year. And I had gotten into engineering schools, and I never looked back. And so engineering became sort of my path. And during engineering school, I actually discovered my real passion for evolutionary biology and molecular biology. And that sort of really sparked my interest in what I currently work on.
How did you get that interest in evolution in engineering school?
Well, I think partly because I was a chemical engineering student without a real kind of rudder about exactly what I wanted to do. I was clear that I could continue doing chemical engineering, but I was getting very intrigued by ideas of philosophy and selfishness and everything from economic markets to biology. And I started reading Richard Dawkins and Ben Lewin's genes, textbooks on molecular biology. What I was missing was some sort of formal guidance as to how to go about sort of thinking about this. And so at the time I was really interested. My faculty advisor in chemical engineering introduced me to this young professor who had just set up shop in the Indian Institute of Technology where I did my undergraduate. And he said, well, you should come to my class, Introduction to Molecular Biology. I thought that was great, except it completely overlapped with the time that I had to take a chemical engineering class, which was like not negotiable. So he said, well, you can't come to my class legally. What I recommend is you come to my office in the afternoon. I'd have given the same lecture in the morning and I can just kind of talk about it in a more informal setting. And, it's sort of, I remember because he was like, he's this amazing, very articulate professor. He was a chain smoker, and literally I must have smoked some packs of cigarettes by secondhand smoke. But over the course of a semester, he really taught me molecular biology, and it was actually like the best way to learn, because one-on-one I could interrupt him anytime. He wasn't really worried about going through all the material that he needed to cover in his class, and so it was that was probably that semester that really changed. I was like, I can do this, I can think about this, and that would be something I could do.
I think that's a great way to teach a course one-on-one. And when I have office hours, I sometimes think about that because you can really get into detail and not worry about time.
Yeah, it was a self-designed course, really. I mean, he asked me what I was interested in. I was interested in oncogenes at that time, and viral oncogenes in particular. And he said, well, let's just think about that. And then he suggested papers that I began to read. I think I alarmed my faculty advisors a little bit because it became clear that I was choosing more and more biology topics. But they were happy that at least I'd found something that I was very excited about.
So if I remember from a previous chat we had, you then went for a PhD in the US, right?
That's right. I applied for PhD programs in India and in the US. But in India, I think the lack of my formal biology background was, in most institutes, considered a significant impediment. It was difficult for me to break through sort of that initial, and there was a lot I didn't know. But I was just very, very motivated to kind of go past that, to say, well, I don't know a lot of stuff, but I'm very interested and I feel like I can catch up. And the one thing, even though standardized tests get a really bad rap, I suspect that I ended up getting a lot of admissions in universities because in the US, standardized tests was really the only way they could evaluate somebody who didn't have a biology background. And my scores were very good. So they said, well, we will... At that time, we had no Skype, etc. So they did a phone interview just to make sure I could speak English, and that was it. And I sort of chose particular people that I was interested in working on based on my limited reading, who were working on mobile elements, because I felt like that was a really good way to encapsulate sort of almost like a pre-stage for a virus, where genes that were jumping around in the genome.
Where did you do that?
That was at University of Rochester in upstate New York, which was interesting because I'd never seen snow before.
I was going to say, big change, right?
Big change. In a way, it was really nice because there were many evenings and winters where there was literally nothing to do, and it sort of was again an amazing. It's a small department, got a lot of attention from my mentor and also from other faculty. And I often wonder, because I advise a lot of students, including in my lab, about where they should go to graduate school. And I felt like, even in hindsight, even though I'm making a virtue out of necessity, it was really good for me to actually have graduated in that department because I felt like I got a really excellent education. This equivalent of this one-on-one mentoring that I got as an undergraduate, I got that as a graduate student, and that was fantastic.
So at this point, I presume you knew that you wanted to have a laboratory of your own?
After I passed my qualifying exam, I felt like I could seriously consider that as a job prospect. The thing that actually really sort of sealed it for me was we had been doing this series of fly genetic experiments, and I realized sort of about 2/3 of the way in that I had made a mistake, and it was going to cost me about 4 months to rectify my mistake. And I remember thinking about this, and I thought, well, I'll just do it over. I'm really interested in knowing, and sort of recognizing that it didn't bother me that I had to go back 4 months. I'd wasted 4 months of my life doing something which was basically going to amount to nothing except some pages in my lab notebook. But I was so excited about the answer and at the experiment, I figured that that's what I should be doing.
Great.
Yeah.
And where did you go for a postdoc?
For my postdoc, I went to the Fred Hutchinson Cancer Center to work with Stephen Henikoff. So I initially applied to Steve Henikoff's lab because he had been pioneering these methods for remote homology detection from proteins. And I think that's one of those technological advances that had really sort of changed the way we think about how proteins evolved. But when I interviewed in his lab, he actually told me about this completely different project that really fascinated me, which led me to think about potential molecular understanding of how species originate, how one species splits into two that cannot reproductively mate with each other, etc. And the thing that I was excited about going to his lab was I had never really done cell biology, and his lab was really fantastic for cell biology. So I sort of learned evolution and molecular biology in my graduate work, and I was intrigued about sort of more explicitly learning cell biology as a postdoctoral fellow.
So at least from my view, your work is computationally intensive. I don't know if you feel that way or not. Is that something you learned as an engineering student or as a PhD student or both?
I actually think that the tools have become so sort of nice that you don't have to be yourself a computational biologist unless you're actually doing the next generation of tools. What I think the engineering background gave me was a very good sense of intuition. And I think that's something that we don't value as much. Like there are some sort of things which are much easier for somebody who has a basic mathematical background to grasp. And it's not that biology students would not get that, it's just not intuitive. And it's just the same way as somebody would pick up like a molecular biology experiment. It was much easier for me to pick up a bioinformatic experiment and understand why some things work versus not.
I think that's an important concept, to trust your intuition. A lot of students don't. They want help to figure out what something means or what's important, but I like that.
Yeah, I feel like, there's this very perilous sort of in-between path. I have graduate students in my lab and I go through, and I sort of always tell them, some of them frequently don't come back after this conversation, but I often tell them that, my role is to help guide them, but I'm not going to tell them what to do on a day-to-day basis. Sure. Because some of the best training I learned while I was a graduate student was learn from my own mistakes, sort of, as long as they were mistakes that you could sort of recover, in this case, 4 months long.
Okay.
But, my advisor, it was not that he didn't anticipate or he was not interested. He was just very interested in me figuring out on my own. And so some of the sort of, it ended up being a 6-year PhD. It could easily have been a 5-year PhD, but that year was like the best education that I ever got.
So you spent a good part of your career working on what you call evolutionary arms races. So first I'd like you to tell us what that means.
So evolutionary arms races actually is a metaphor that was coined by Lee Van Valen, who was one of the sort of the pioneers of the field of Red Queen conflicts. The idea being that when two competitors are competing with each other for some sort of fitness space, let's say host versus the pathogen, the arms race part comes because what is in the best interest of the host is often not in the best interest of the pathogen. So they're often actually seeking mutations or changes that will actually help themselves keep pace with this sort of very rapidly changing competitor, because as soon as they stop doing so, they run the risk of being driven to extinction by the relentless nature of the adaptation that's going on for the competitor. So in a way, it's very much like how Darwin anticipated adaptation, but he anticipated this in a much more static sense, in the sense that you have adaptation as you transition to a new ecological niche or to a new island. And what Red Queen interactions sort of made it clear was that there was never going to be a universal optimum. It was completely dependent on who your competitors were in the population. So there's no such thing as a universally good fitness change, as there might be in the course of adaptation for, let's say, better higher, taller trees or better photosynthesis. This was a case where your fitness space was really determined by your genotype, but also what the pathogens in your environment were doing. And so that was a fascinating kind of concept.
How did you get interested in this?
I became interested in the idea. I think I got interested in it from reading Dawkins. So I sort of give a lot of credit to the value of really excellent science writing, where I began to really appreciate that in a lot of biology, I had been brought up to think about biology as this, seeking this optimum. Cell biology, the components of the cell are working in concert with each other to reach some optimal fitness or state for the cell. And I realized that the evolutionary viewpoint was there was no such thing. It was sort of at best a transient optimum, and then it would quickly be dispensed by some sort of cheating or selfish behavior by a cellular component, and then the cell would have to reach this optimum completely on its own, de novo. And so this relentless sort of changing of sort of these optimum states was really fascinating. Sort of a good analogy is that if you actually have a universal optimum, then you could basically take, let's say, parts from different Swiss watch manufacturers, interchange them, and they should work perfectly well. But because they're each sort of driven by their own sort of interests and their own sort of competitive interests, you cannot actually fit the gears of different watch companies together. In fact, if you do so, you basically have a non-functioning watch. And then it's very much like what would happen with the cell that's now faced with these, newly evolving components. And it became fascinating to me to understand how a cell works, not because of these interactions, but in spite of these interactions. And so that was the sort of driving and pathogens became like the biggest sort of corkscrew in the works in a way, because they were constantly challenging basic cellular components.
So the Swiss watch analogy brings to mind people who are trying to do synthetic biology now, right? It's almost, if you make a chromosome, whether it be a viral or yeast, it may not work as well as the naturally evolved one.
It's a really excellent point. I feel like people are anticipating as much as they can anticipate. But some of the best experiments, for instance, introduction of a virus to eliminate Australian rabbit populations that have told us is that nature is much more insidious than we realize.
Right.
There are many more outcomes than we can even anticipate, even with all the science fiction that we read. The outcomes that emerge from nature are amazingly divergent from what our expectations might be. It's completely possible that synthetic biology might design this billion-dollar organism that's designed to do biofuels or some sort of remediation of some sort of ecological states. But there's always a risk that organism is going to, once faced with Darwinian pressures, jettison this billion-dollar investment in its molecular biology and basically pick up completely new things from the environment. And suddenly what happens, so I feel like the jury's still out whether this is going to work in the long term. It's a fantastic idea in terms of when you can control, but even if you inject these synthetic organisms into the human body, that's it's as ecological as like going out into the woods and picking moss from trees, because there's a lot of genomic diversity that has the potential to influence these kinds of experiments. This is not to say the experiments aren't valuable,
Sure.
but it's unclear whether the evolutionary success of these experiments is going to be exactly what was designed.
Now, I think we have to do them, but we can't assume they're going to work exactly as we plan, right?
Yeah.
So can you tell us, in this field that you're working in, arms races, I'll call it, what is a key experiment that you would say characterizes your laboratory?
So I think one of the first key experiments that was done in my lab by a young postdoc in my lab, Sarah Sawyer, whom you've met before, in collaboration with Mike Emmerman, who's one of my colleagues. And we became very fascinated with the idea of focusing not on what is conserved in protein evolution, which has basically been really the focus of molecular biology. What are the residues of enzyme that are conserved through evolution? What has evolution preserved or natural selection preserved? We became interested in what are the parts of the protein that really look like they've gone to hell in terms of their evolutionary conservation, because we reasoned that those very rapidly evolving parts of the protein might be the key to these very rapidly evolving lock and key type interactions that viruses and hosts might be manipulating. So just at the same time, this was very close to when I was starting my lab, so it was very kind of almost designed in a way this way, because Michael Malam's lab had just discovered ApoBex as this new restriction factor for HIV. And Joseph Sadrowski's lab had discovered TRIM5 alpha as one of the very key restriction factors for HIV. But he had also discovered that TRIM5 alpha was not universally good, which means that the rhesus version of TRIM5 alpha helped defeat HIV, but the human one did not. And we became fascinated by the fact that here are two relatively closely related proteins that have very different outcomes in the context of this, retrovirus. And so Sarah basically, using the evolutionary insights from my lab and really the HIV expertise from Michael's lab, really set about to test the hypothesis that, in fact, if we focused on just the rapidly evolving residues in TRIM5 alpha, it actually turned out to be less than 10 residues, most of them clustered in one very sort of tight loop. She predicted she could switch specificities just by switching these rapidly evolving domains. And that was like the eureka moment that indeed turned out to be the case. So really, whereas the infrastructure of the protein or the nitty-gritty mechanistic aspects of the protein were indeed very well conserved, what made the protein able to recognize different viral targets actually turned out to be very rapidly evolving. So in a way, I feel like that's a really key experiment, because it was the first proof of principle, and we've now demonstrated this for a number of different antiviral genes, that really by focusing on what is rapidly evolving, or what we refer to as positively selected, we can actually get a very good estimate of what is the viral host interaction interface.
So that key experiment has led to other similar investigations.
Exactly. Yeah. So, and in fact, this paradigm has allowed us to sort of really take one step forward, because I felt like people were studying viral evolution. There were some fantastic people studying this, and then of course there were people studying the molecular and cell biology of host-virus interactions. But there was like almost no overlap between those worlds. And what Sarah's paper basically demonstrated was this purely evolutionary guided hypothesis testing approach was a really great way to kind of jumpstart this investigation. And that's what has, that has become quite well adopted now in the field.
Besides using viral systems in your lab, you also use other non-viral systems to get at the same question, right?
That's right. We are very interested in the idea of what drives this innovation, what the genetic innovation, both in terms of rapid amino acid changes, but also genetic innovation in terms of birth and death of genes for chromosome processes that we care about.
Is there a big unanswered question that looms above you that keeps you awake at night?
So the thing that really sort of drives me is still this idea of Paleo Biology. So while it's really great to actually be able to test hypotheses with existing viruses and sort of averaging evolution over the past millions of years, I'm intrigued by the idea of how much of our existing biology is really driven by our sort of episodic interactions with viruses, many of which don't even exist today because they've gone extinct. And some of these viruses have left imprints of their fossils in our genome, and we can try to resurrect them, and we have done so. But many of them have not really left traditional fossils, so there are no viral remnants in our genome, but nonetheless, their influence on the host genes have basically been inherited by everybody, including us. And that influence is something that I'm very interested in, whether a virus sort of drove an evolutionary path down one landscape that forced us down a path which is actually suboptimal based on a current day version of a virus that we might encounter. I'm intrigued by this paleovirology, and I'm excited about the prospects of what we can do, because our ability to detect this remote homology, these sort of dead viral fossils which are getting more and more decrepit with every passing million years, we are actually getting better at being able to kind of pick them out from the sort of our genomic DNA. And I'm excited about what those stories are going to be. Ultimately, evolutionary biology, a lot is about the discovery of these very interesting stories.
The paleovirology you talk about is really old, millions and millions of years old.
Yes.
But so something like a 30,000-year-old virus probably isn't much interest to you. I'm thinking about the recent one from Siberia.
Absolutely. I think it's very interesting. I think what's nice is that it enables us to actually calibrate whatever metrics we use, and having something which was definitely operational, like smallpox, etc., within human history is really nice for our calibration tools. That is also paleovirology, except we are very interested in paleovirology even deeper.
Right.
And the deeper we can go, I feel like the more powerful the approach is.
Can you give us an idea of how technology has changed? Now, I think you are among the youngest of the people I'm talking with, so your view may be somewhat compressed, but have you seen things change that have really helped your field in a big way?
Not my field in particular, but I have been just amazed by how sequencing technologies have sort of changed the way we think about even sort of routine genetic experiments. So viral genetics, we are able to isolate in high detail and almost in the absence of our ability to culture some of these viruses, are able to still make very, very nice biological inferences just by the sheer power of us being able to sequence these viral genomes and viral nucleic acids from the environment. And I think that has really kind of revolutionized the way we think, and even the types of things that we think about. I'm a little concerned that there's this sort of, every time there's a new technology, everybody sort of jumps on it, and whether everybody's using them for intellectually stimulating purposes. And I think we are not quite at the point yet where everything intellectually unstimulating has been culled out. But I think that every time there's this very big technological leap, people realize more and more what are the amazing things that can be done. In a way, I feel like that's something that is still untapped, amazingly. There's a lot we can do with these technologies, which just is still not quite at the point where we are intellectually able to fully take advantage of these technologies.
Do you think a new technology is developed to solve a problem, or you think the technologies arise and then people find applications for them?
I'm 100% confident that the technology was actually developed to deal with the clearly developing market about human genomics, because the scale of the human genome presents a challenge, and to make it cost effective. And there has been advances in terms of the human genomics, and the ability to detect mutations, and human genetics has really advanced by this. But there's almost been like 100-fold increased advance in microbial genomics that has emerged from these technologies. The sheer power of being able to assess 10 billion reads in one experiment has allowed you to sort of, it's made you in a little way sort of lazier in terms of your hypothesis testing, but in another way it actually allows you to really get a very good grasp of the complexity of the problem before you have the ability. So people have been able to use these to get very detailed mutational landscapes for things like influenza virus and polio. And it's not that we would not be able to have intellectual advances in the absence of the technology, but the presence of this data just is a game changer in terms of how detailed your estimates can be.
So our current DNA sequencing ability really was motivated by doing the human genome, right?
I think the 1,000 genome sort of really propelled the challenge that we needed to sequence the human genome within $1,000 really propelled this sort of cost drop.
And then it was, the microbiome was really discovered because of that.
Absolutely.
And all the influenza evolution is based on that technology.
Once the technology was different, everybody from ecological sort of minded people to microbial minded, they were all on it. And in fact, their advances far exceeded what we've actually learned from the human genome.
It's amazing. It's just stunning. I wonder what's next.
Yes, I'm hoping it'll get cheaper because it's an expensive, it's powerful but very expensive. And if you're not careful, you can actually spend a lot of money before even realizing kind of where your budget went.
Yeah. So if you look back on your work, and I realize that you're pretty young in your career, can you point something out that you think has made the biggest contribution to science in general?
I feel like, I don't actually have the perspective to sort of say this in an explicit way, but as we go to ASB meetings and stuff, I feel like this is not, of course, my own contribution, but we are seeing a greater synergy between what used to be very explicitly evolutionary approaches in meetings for American Society of Virology, American Society of Microbiology. And some of this has come about because this new data has posed completely new challenges, which evolutionary biologists know actually how to deal with. But I think even more so, I think is the fact that people are recognizing that this evolution-guided approach is a very good complement to more traditional approaches. Intellectually, I feel like that's the biggest advance. But I would say that even though I haven't had so many alumni from my lab's only about 11 years old, I suspect, when the dust settles, the biggest contribution that my lab would have made would be the people we've trained. Because even some of the people who left recently enough that they're still sort of junior faculty are making significant impacts on their communities.
So if you hadn't become a scientist, what would you have done?
Actually, this one is very easy for me. So even though I was very interested in becoming a doctor, I always wanted to be a novelist. I felt like I could write well. Some of the people who criticize us in our reviews sometimes claim maybe I have too much fiction in my papers, but I feel like there is a certain charm of creating a story to sort of bring people interested. And I think evolutionary biology in its richest form is about those stories, about what were the conditions that created a particular mutational scenario. And I always felt like I would be a good writer. Actually, it turns out I was not a very good writer, but I was at least always interested in being a very good writer.
Would it be science fiction that you would write, you think?
I was very interested in science fiction, yes.
Yeah, but you haven't written anything?
Nothing that is worth talking about.
So you should stick with your current job, right?
Yes, I think I'm more successful in my current job.
So a lot of the readers of this textbook and people viewing this video are thinking of a career in science. Do you have any advice for them?
I would say that, when I was getting into the business, even though I came from a different field, there was this thought that, a lot about molecular biology that needed to be discovered had already been discovered. And then there was this wave where it became clear that small RNAs had a huge influence, which had not really been anticipated. And suddenly that changed and answered so many long-standing mysteries and changed the way we perceive even basic things like genetic and epigenetic inheritance. And yet the roots of those discoveries are in sort of very run-of-the-mill, if you will, sort of observations, which led people down this very sort of long path of discovery that just sort of changed the world. I would say that the CRISPR system, which is like going to revolutionize our abilities to do genome editing, again owes itself in some ways to a very important discovery in bacterial genomes as a mechanism of defending against viruses, once again, as a red queen interaction. And yet, if you go back 10 years, there was basically nothing about this in the works. And so I feel like there's always a temptation to think that we are really at the cusp of it and there's nothing more on the horizon. But really, if you go back every 10 years, there are amazing discoveries. And those are mostly discoveries made by students working in labs that are not sort of goal-oriented with the idea of discovering a genome editing enzyme, but it's about basic discoveries. And ultimately, I think what I felt like once I reached peace was when I discovered that I found something I was good at, and it's actually something that I could make a career out of. And I feel like students often don't sort of pay a lot of attention to, is this really exciting to them? How do they deal with success, but also how do they deal with failures, and do they really enjoy the process of discovery? And I think if you do enjoy the process of discovery, I think, despite sort of what people have said recently about the state of biomedical funding in the US, et cetera, I have to say it's still the best possible job And I think this career rewards you. It certainly punishes you, but the rewards are far greater than what you can imagine.
I've been speaking with Harmit Malik of the Fred Hutchinson Cancer Research Center. Thanks so much for talking with me today.
Thank you, Vincent.